Hello everyone, welcome to the ITLS Academy. Today we are going to discuss the topic that is your determination of total carbohydrate, and the method that we are going to use is a phenol sulfuric acid method. Okay, so we are using the method that is your phenol sulfuric acid method. We all know that this carbohydrate play a very essential role in providing the energy. Okay, play a very essential role in providing energy. It is the good source of energy. Okay, we all know that 1 gram of carbohydrate will give a 4 kilocalorie. Okay, so this carbohydrate is also a main nutrients in the nutrition. Okay, or you can also say that it is the most important part of the food. So for this purpose, we have to determine its concentration or its amount in a food product. Or here, the method that we are going to use is a phenol sulfuric acid method. See, determination of the carbohydrate is basically like it is determined by two types. One is your qualitative and another one is your quantitative. Okay, now qualitative means that either the carbohydrate are present or not, and quantitative means that the carbohydrate are present, but we don't know how much amount of carbohydrate are present in a particular food product. So here what we are going to do, we are going to define the quantity of the carbohydrate present in a food product, and the method that we are going to use is a phenol sulfuric acid method. Now come to the next one, that is your introduction part. Okay, so we all know that this phenol sulfuric acid method is what it is, very simple method and very rapid, or you can say that very easy method, and very rapidly of the result of this method are come. And it is also known as a colorimetric method, which is used to determine the total carbohydrate present in a sample. Okay, it can also detect all type of the carbohydrate present in a food product, like your monosaccharide, disaccharide, oligosaccharide, and polysaccharide. Okay, but it uh, it will detect all type of carbohydrate, but their absorbability of the different carbohydrates are vary. Okay, so unless a sample is known to contain only one carbohydrate, the result must be expressed arbitrarily in terms of one carbohydrate. Now see what is written in the slide. The phenol sulfuric acid method is a simple and a rapid colorimetric method. Okay, it is what? It is a simple and rapid colorimetric method which is used to determine the total carbohydrate in a sample. Here the sample of what? Sample is a food sample in which the carbohydrate are present. The method detects virtually all classes of the carbohydrate, including mono, di, oligo, and polysaccharides. Although the method detects almost all carbohydrate, the absorptivity of the different carbohydrate vary. When the absorptivity of the carbohydrate vary, then the result also will be different. Thus, unless a sample is known to contain only one carbohydrate, the result must be expressed arbitrarily in terms of one carbohydrate. Okay, now come to the next one. We all know that when we using this sulfuric phenol acid method, so in that case, what we are going to do, we are using the concentrated sulfuric acid, and this concentrated sulfuric acid break the polysaccharide, oligosaccharide, and disaccharide into a smallest chain that is called a monosaccharide. Okay, so here the pentose, that is 5 carbon carbohydrate, they are what? They are dehydrated to a furfural, and hexose, which is 6 carbon compound, it will dehydrated into a hydroxymethyl furfural. Okay, so these compound when react with the phenol, they produce what? Yellow gold color. Okay, and for product whose in which xylose values are high, like your wheat bran, corn bran, or xylose must be used to construct the standard curve for the assay, and it should be measure the absorption at 480 nanometer. Okay, and the product in which the hexose sugar level are high, then in that case the example are what? Glucose, which is commonly used to create the standard curve, and this absorption are measured at 490 nanometer. Okay, we all know that carbohydrates are what are the major sources of some caloric product like your soft drinks, beer, and fruit juices, and it will supply 4 kilocalorie per gram of carbohydrate. That I already told you. If we consume 1 gram of carbohydrate, then it will give 4 kilocalorie. Now see what is written in the slide. In this method, the concentration sulfuric acid break down any polysaccharide, oligosaccharides, and disaccharides into monosaccharides like pentose, your 5-carbon compound. Then they are dehydrated to a furfural, and the hexose 6-carbon compound to hydroxy methyl furfural. This compound then react with the phenol to produce a yellow gold color. Okay, so this is very important in the point of the MCQ, that when the compounds react with the phenol, then which color they develop, they will produce a yellow gold color. For products that are very high in xylose is what? Xylose are a pentose, means that they have a 5-carbon compound like your wheat bran, corn bran. Xylose must be used to construct the standard curve for the assay and measure the absorption at a 480 nanometer. For products that are high in hexose sugar, now in that case glucose is used to create the standard curve, and the absorption is measured at 490 nanometer. Carbohydrates are the major source of calories in soft drinks, beers, fruit juices, and it will be supply 4 kilocalorie gram per carbohydrates. Okay, now come to the next slide. Objective is very simple. What we are going to do, we have to determine the total carbohydrate in the sample, and here the samples are what? Samples are a soft drinks and the beer. They are what? They are a sample. So what we are going to do, we are going to determine the total carbohydrate content by using the sulfuric phenol acid method. Now the principle are what? Carbohydrates. Principle behind this experiment are what? This carbohydrate react with the uh, strong acid, and when they react, they produce a heat. Okay, then they condense with the phenol and form a yellow gold compound, which measure into a with the help of the spectrophotometer. Now see what is written in the slide. Carbohydrates, like your simple sugar, oligosaccharides, polysaccharides, and their derivative, they react in the presence of strong acid and heat to generate foreign derivatives that condense with the phenol to form a stable yellow gold compound. Okay, that can be measured spectrophotometrically. Now the next one is your chemical that we use in this experiment. We are using 3 chemicals here. One is your D-glucose, then the second one is your phenol, and the third one is your sulfuric acid. Okay, so D-glucose, whose chemical formula is C6H12O6. Its case number is 50-99-7, and the hazard, it does not contain any hazard. Phenol, whose chemical formula is C6H6O, case number 108-95-2, and the hazard related to this chemical is what? They are toxic in nature. So we have to avoid to come in contact with the phenol. And the next one is your sulfuric acid. Sulfuric acid, whose chemical formula is H2SO4. The case number is what? 7664-93-9. The hazard related to this sulfuric acid is what? They are corrosive in nature. So these are the chemicals and the hazard associated with this chemical, and these chemicals are used in this experiment. Now the next one is your reagent, the reagent that we use in this experiment. The first reagent is your glucose standard solution, and this glucose standard solution are prepare. We have to weigh 100 milligram of glucose and just divide into a 500 ml of distilled water. Now when this glucose are completely dissolved into 500 ml of distilled water, then the remaining 500 ml of distilled water we are going to add into it, and the solution will become a 1000 ml, that is your 1 liter solution. So here the standard solution of the glucose will be prepared 100 milligram per liter. Now the second one is that we have to prepare a phenol 80% weight by weight in H2O 1 ml. For this, what we are going to do, we take a 20 we are take a 20 gram of deionized distilled water into 80 gram of redistilled reagent grade, that is your phenol. Okay, we take 20 gram of deionized distilled water, or you can say that 20 ml, and in which we are going to add a 80 gram of redistilled reagent grade, that is your phenol crystal. Okay, now the next chemical is what? Your sulfuric acid, and the sulfuric acid is in the concentrated form. This is very important. The sulfuric acid that we are going to use in this experiment will be in concentrated form. Now the next one is your hazard and precaution and waste disposal related to this experiment. We all know that we are using a concentrated H2SO4, and along with it we are also using the 80% of phenol solution. So these two solutions are very crucial, and when we are handling this, we have to take a care before handling it. So the we have to wear a gloves and even the safety glasses in all the time, and the other laboratory practices that are very essential, we have to follow it. Okay, so we all know that this concentrated H2SO4, that is your sulfuric acid, are very corrosive in nature. So we have to avoid it to come in contact with our clothes, shoes, or skin. And when we are discarded this phenol, they are toxic in nature. So when we are discarding it, we have to discard it in a hazardous waste. Okay, and the another waste which contain the phenol, in that case we have to drain out by using the water rinse, and the other good laboratory practices we have to follow, which is good for the environmental health, and also the safety protocol which are given by the university or the institution. We have to follow it. Now see what is written in the slide. Use concentrated H2SO4 and the 80% phenol solution with caution. Wear gloves and safety glasses at all time, and use good lab technique. The concentrated H2SO4 is very corrosive. Example to clothes, shoes, and skin. The phenol is toxic and must be discarded as a hazardous waste. Other waste not containing phenol likely may be put down the drain using a water rinse, but follow good laboratory practices outlined by our environmental health safety protocol at your institution. Okay, now the next one is your material that we use in this experiment. So the first material that we required is a beer. Beer is what? Beer is the sample. Okay, so lit and regular of same brand, bottle to collect the waste, cuvette for spectrophotometer, aluminum flask of 100 ml for a distilled water, two aluminum flask of 500 ml for the beverages, gloves. Okay, then we also required the mechanical adjustable volume of pipette of 1000 microliter, 100 microliter, or 200 microliter with a plastic tips. Pasteurized pipette and bulb, parafilm, pipette bulb or a pump, repipettor for fast delivery of 5 ml of concentrated H2SO4. We also required a soft drinks, clear color, dyed color, regular of same brand. This soft drink is also the sample in which we are going to determine the carbohydrate concentration. 20 test tubes, 16 to 20 mm internal diameter, test tube rack, 4 volumetric flask of 100 ml to 2 volumetric flask of 1000 ml, that is your 1 liter. Okay, so we required around 6 volumetric flasks, in which 4 volumetric flasks are for 100 ml and 2 for a 1 liter. We also required a volumetric pipette of 5 ml and 2 volumetric pipette of 10 ml. So here also we required 3 volumetric pipettes, 1 of 5 ml and 2 of volumetric pipette of 10 ml. Now the next one is your equipment required for this. We required only 3 equipment for performing this experiment, like your spectrophotometer, vortex, and water bath, which maintain the temperature 25 °C. Now the next one is your procedure. Okay, so first of all, what we do, we require a standard curve tubes. Using the glucose standard solution, add double deionized distilled water as indicated in the table below. According to this, what we are going to do, we are going to add a deionized distilled water. Pipette allocate of the glucose standard into the clean test tube, duplicate for each concentration, such that the tube contains 0 to 100 microliter of glucose using a 1000 microliter mechanical pipetter or to a pipette the sample. In a total volume of 2 ml, this tube will be used to create a standard curve with the value of 0 to 100 microgram of glucose per 2 um, per 2 ml of the 0 microgram of glucose per 2 ml sample will be used to prepare the reagent blank. So here the ml of glucose stock solution, and here the amount of microgram glucose per 200 ml. So we and this is your ml of double deionized distilled water. This is your 0 microgram, 20 microgram, 40 microgram, 60 micro, 80, 100 microgram. So when we are taking 0 microgram of glucose solution, so in this case we are not going to add glucose stock solution. Okay, so when we are adding 0.2 ml of glucose stock solution, then in that case we are going to add a 20 microgram of glucose of 2 ml, and in which uh, in this solution we are also going to add a 1.8, no, sorry, this is a this column is for a standards of microgram of glucose per 2 ml. So for this we have to prepare a 20 microgram of glucose, 40 microgram, 60 microgram, 80 microgram, and 100 microgram. Okay, so when we are taking the first one, in that case we are not going to add a stock solution of glucose. So in that test tube we are going to add a 2 ml of deionized distilled water. Now in the second one, when we have to prepare a 20 gram of glucose per 2 ml, then in that case we are going to add a 0.2 ml of glucose stock solution and 1.8 of ml of deionized distilled water. Then when we have to prepare a 40 microgram of glucose solution, then in that case we are going to add 0.4 ml of glucose stock solution and 1.6 ml of deionized distilled water. Then when we have to prepare a 60 microgram, then in that case we are going to add 0.6 ml of glucose stock solution, and then we are going to add 1.4 ml of deionized distilled water. And when we have to prepare a 80 um milligram of glucose solution, then in that case we are adding 0.8 ml of glucose stock solution and 1.2 ml of deionized distilled water. And when we have to prepare 100 microgram, then in that case we are adding 1 ml of glucose stock solution and 1 ml of deionized distilled water. By this way we have prepared the uh, mic mm, microgram glucose solution per 200 ml. Now after that we have to record the caloric content from the label. You will analyze for total carbohydrate content. First one is your regular and diet soft drink of the same brand, and the second one is regular and the light beer of the same brand. So we taking two sample. One is your regular and the diet soft drink of the same brand, and the regular and the light beer of the same brand. Now before you proceed with the same preparation and analysis, record the calorie content on the nutrition label of the sample you will analyze. So you, before starting the experiment, what we have to do, we have to note down the caloric value. Now the third one is your decarbonated the beverages. In this step, what we are going to do with the beverage at room temperature, pour approximately 100 ml into a 500 ml of aluminum flask. Now we have to shake it first, and we have to avoid the formation of the foam in the sample in case of beer, and it will be continue gently shaking until no observable carbon dioxide bubble appear. If there is any noticeable suspended material in the beverages, then filter the sample before analysis. Now we have shake until all the carbohydrate carbon dioxide bubble should be disappear, and if any suspended material are there in the beverages, then we have to filter it before the analysis. Now the fourth one is your sample tube. So the sample tested will contain 20 to 100 microgram of glucose per 2 ml. The dilution procedure and the volume to be assay are given. So this is your soft drink regular diet and beer regular diet. 1 is to 2000 and 0, 1 is to 2000 and 1 is to 1000, and the volume essay for each for 1 ml. Now the next one is your pipette 500 ml of beverages into 100 ml of volumetric flask. Now what we are going to do, we take 5 ml of beverages into 100 ml of volumetric flask and dilute it with the deionized distilled water. Then we have to seal it with the paraffin and mix well. The dilution is 1 is to 20. Then pipette 1 ml of 1 is to 20 diluted beverages into a 100 ml of flask. Then again we dilute it with the deionized water, and then we seal the flask with the paraffin and mix well. Then after dilution, we pipette 1 ml of sample into a test tube, and in which we added again 1 ml of deionized distilled water and analyze each diluted sample in a duplicate. Now after that we are going to add phenol, that is called the phenol addition step. Now in each test tube from part 1 and 4, which contain a total volume of 2 ml, in that case we are going to add 0.05 ml of 80% of phenol. Mix on a vortex test tube mixer. Okay, then when we add this 80% of phenol, after that we mix it into a vortex. Then after that we add H2SO4. That step is called as sulfuric acid addition. Now each test tube from part 5 add 5 ml of H2SO4. The sulfuric acid reagent should be added rapidly to the test tube. Then the direct stream of acid against the liquid surface rather than the against the size of the test tube in order to obtain a good mixing. Okay, these reactions are derived by the heat produced upon the addition of sulfuric acid to the aqueous sample. Thus, the rate of the addition of sulfuric acid must be standardized. Now, we have to use a vortex for the mixing. After that, we have to leave the test tube for a 10 minutes, and then it will be placed into a water bath at 25 degrees Celsius for a 10 minutes. That is to cool them to a room temperature. Now vortex the test tube again before reading the absorbance. Now after that, what we are going to do, we take a absorbance by the spectrophotometer. Now, reading the absorbance, we have to wear the gloves to pour the sample from the test tube into a cuvette. Okay, when we are pouring the sample into a cuvette, we have to wear the gloves because of the sulfuric acid and the phenol compound. Do not rinse the cuvette with the water between the sample. In that case, we are not going to rinse it with the water. Zero the spectrophotometer with the standard curve sample that contains 0 microgram of glucose per 2 ml. That is your blank. With the help of the blank, we measure the spectrophotometer value till the 0 and retain this blank sample in one cuvette for later use. Now, after that, we have to read the absorbent at a 490 nanometer. Read your standard curve tube from a low to a high concentration, that is 40 microgram per 2 ml to 100 microgram per 2 ml. Okay. And then read your beverages sample to be sure that outside of the cuvette are free of moisture and smudges. Wipe the outside of the cuvette with a clean paper wipe prior to insertion it into the spectrophotometer for the reading. Now, absorbance spectra use one of the duplicate tube from a standard curve sample with an absorbance reading 0.5 to 0.8, and we have to determine the absorbance between 450 nanometer to 550 nanometer by reading the test tube 10 nanometer interval. Zero the spectrophotometer with the blank at each 10 nanometer interval. Now summarize your procedure and result for all the standard, and see, this is your this is your data and calculation step. Data and calculation step. Okay, the procedure part is completed. Now, after that, you have to summarize your procedure and result into a standard and sample in that table. This table is used for this purpose. Here, the sample identity. First one is your blank, then the standard 20 microgram, 40 microgram, 60, 80, 100. This is your absorbance at 490 nanometer. And we are taking two absorbance for each sample, and after that we take the average. Now, this is your soft drink regular whose absorbance should be taken at 490 nanometer and microgram glucose 2 ml. Then dilution scheme, then glucose equivalent, that is your microgram per ml original sample and gram per liter of original sample. By this way, you have to measure the absorbance of your sample. Okay. Now, this is for the previous one is for the soft drink, and this is for a beer. Now, sample calculation for the soft drink regular, that equation for line y is equal to 0.01 1x and plus 0.1027, y is equal to 0.648. By solving this, we get the y and x is equal to 49.57 microgram per 2 ml. Now we are using the formula, that is C1, Ci is equal to Ca, V2 by V1 and V4 by V3. And this equation are get from a solution preparation. If you have any query for this equation, you go and see the video which are solution preparation. Okay, concentration C. Okay, now put all the values, then we get a final concentration, initial concentration, that is your 99.14 gram per liter. Okay, now after that we have to construct the curve for your total carbohydrate, and then we have to determine express in term of glucose. Then we have to determine the equation of the line for the standard curve, then calculate the concentration of glucose in your soft drink sample and beer gram, and we are calculating in terms of gram per liter and gram per 12 FLOZ. Okay, note that 29.56 ml is equal to 1 FLOZ. Then we have to calculate the caloric content based only on carbohydrate content of your soft drink sample and beer sample in term of calorie per 12 FL.OZ. So this is the table for putting the values, that is your soft drink, regular diet, beer, regular diet, the concentration of glucose per 12 FL dot OZ, measure calorie per 12 FLOZ and the nutritional labels. Then plot the absorbance spectra obtained by measuring the absorbance between 450 and 550 nanometer. Now we are taking the absorbance between a 450 to 550, that is 450, 460, 470, 480, 490, 500, 510, 520, 530, 540, 550 each in each absorbance. Then after that we plot a graph. So this is all about the determination of the carbohydrate by using the sulfuric phenol acid method. Thank you for watching. Please like, share and subscribe. If you have any query, just let us know in a comment box. You can also ask your query to the helpline number that are already mentioned in your slide. You can also visit our website, that is www.itlsacademy.com, for further inquiry. Follow the ITLS Academy on different platforms like Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and also you can also subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. Thank you so much. If you have any query, just let us know in a comment box.